Hello, my name is Cormac Coughlin, and this is my dad, Charles Brian Coughlin, who is a senior lecturer at the University of North Florida for biology and a forensic biologist. Do you consent to being recorded and uploaded to YouTube and emailed to my teacher?
Yes, I do.
So this interview will be about the switch to DNA evidence. How difficult was it to switch from what was perceived as normal crime investigation at the time to DNA evidence?
All right, so I need to give a little background there. Forensic biology kind of started with the idea that biological material could be left behind at crime scenes that would help link possible perpetrators to the crime scene. There's a basic concept called Daubert principle in crime science, that you have to establish that victims and attackers or perpetrators were in the same place at the time of the crime, or could have been at the same place at the time of the crime. And that Daubert exchange principle suggests that materials, whether it just be physical evidence or biological evidence, could be exchanged at that moment of the crime. And so up until the time I got involved, this was often done with protein testing in blood. And so at the time, forensic biology had a different name. It was called forensic serology, which refers to the blood. And so they do different types of blood typings, for example. But the time I entered in the field in 1995, they were doing something called restriction fragment length polymorphism, RFLP for short. And that was a way of analyzing the DNA found in body fluids that would be left behind at crime scenes and matching them to candidates or possible contributors in the crimes. And this was a technique developed in England by Sir Alec Jeffreys, had been applied there, was well known, and had crossed the Atlantic and taught to the biologists in the Federal Bureau of Investigation, the FBI. And thereon was disseminated to different state laboratories. And so the man who taught me to do restriction fragment length polymorphism testing on DNA was trained by the FBI, who was trained by Jeffreys. And in that science, we would take whatever DNA was present, so it could be a blood spot, say, extract the DNA, which is the genetic material from the cells, and then we would had a way of typing the alleles to discover what certain parts of the DNA had codes specific to individual who contributed them. And so these particular alleles would be found in all DNA types, but were variable. And so that allowed us to kind of statistically note how frequently you would see the DNA in the human population. Particularly in this case from America, as the FBI had a DNA database constructed showing the frequency of these DNA alleles. So, for example, one allele might be seen in the human population in one person in 10,000 persons. And so if you saw that allele present, you would know that it was specific to one person in 10,000. And if you test a series of these, say you had a second allele that would be found in 1 in 5,000 persons, then statistically you can determine the likelihood of seeing these combinations of alleles in populations, and often that would be a very high number. So it might be, you'd see 1 in a million or 1 in 2 million persons would have this particular sequence of alleles. And that was convincing evidence in court, because you could analyze DNA like a blood spot in a murder case that may have been the perpetrator's, the attacker's DNA, and say, yeah, I see this allele set from this blood spot, and you would see its frequency of occurrence in the United States would be 1 in a million. And by the way, it matches the defendant's blood. And that's usually pretty convincing evidence. But at the time I started doing this work, a revolution in DNA analysis occurred. A new technique called polymerase chain reaction was developed that could amplify whatever DNA was there and produce a similar amount of information on the alleles. And so instead of being limited to testing only spots that were the size of a quarter or larger, you could test almost invisible spots, or DNA left behind that you couldn't see the cells that they existed in. So you could swab a doorknob and get a DNA type of the doorknob. So it just revolutionized forensic biological crime science, because now we could test almost anything and demonstrate that the cells from a person were left behind at a crime scene.
How was the general public's perception of this at first and going onward?
Well, as you can see, it's kind of complicated because it involves an understanding of biology and DNA. And so I think that there was first a lot of suspicion about it. And certainly I would say that it was viewed with some suspicion of its being used as an evidence of technique. But eventually, because of popular crime television shows like CSI, it came into those plot lines and people heard of it and it became accepted in the culture. It was actually much harder to get it accepted in the courts because the courts kind of lag behind public opinion, and there's the process of getting it accepted in court as something that produces viable, real evidence in crime, solving crime. And so there was hearings called the Daubert hearing and the Fry hearing that were needed, where essentially the evidence and the way it's gathered and the people who gather it, those things are on trial. And these hearings decide whether it can be used to help solve the crime or not. And so the Daubert hearing, or standard, is that the person who is providing the evidence in court is an expert. Are they an expert enough in their area, so in my case in biology, to be able to render an opinion about the case or within the case of in that particular technical area? And so that's a different standard than is typical, because usually witnesses are only allowed to testify to facts. But an expert witness can render an opinion about the facts if they are respected and pass the Daubert standard. And so they have to have a certain level of credibility and experience for that to happen. And then in the Fry hearing, that's to whether or not this technique that we would discuss, particularly the new one like PCR, is it viable scientifically? Does it follow a protocol? Is there an understanding in the scientific community and by scientists that there's a consensus that it's true, that it actually works and does what the expert witness says it does? But if any new technology in crime science can get through those two standards, it can be used in real caseworks and trials. And so that was what was necessary to get this type of evidence in the time I was working on it into the courtroom. And so several times usually my credibility would be attacked on the stand, so that the defendant and the defendant's lawyer was trying to show that I wasn't a credible expert. And then if that didn't work, if I got established to the jury and judge as a credible expert, they might try to attack the actual test itself. And so that made some of my testimonies longer and more difficult.
How did you get the jury to understand what you were testing? Because at that time it was really complicated.
Yes, it was. I depended upon the prosecuting state attorneys for this most of the time, because as a witness on the stand, you don't get to say whatever you want to say. So it wasn't like I could just get up on the stand and give a lecture about DNA and DNA testing. So you depend upon the state attorney to ask you the right questions. And a good state attorney knows what to ask so that you can educate the jury. And remember, we're talking at a time where DNA was only mildly understood, and the whole process of testing DNA back in the 90s was not well understood by the general population. And those are the people, those are the citizens that make up the juries, the average individuals. And so you had all different educational levels. Some people had no high school or little high school. And so it was a challenge. So typically the prosecutor would ask enough questions that allowed me to educate the jury about biology and DNA, but not go too far to make it something they couldn't understand, but just get across that we understood what DNA was, could communicate to the jury, and then explain how we would test that DNA. Sometimes it depends on the people making up the jury. They wouldn't accept it. To them, what's more important is witness testimony. What did the victim say? What did the alleged perpetrator say? That was more important to them. But usually people, once you got credibility as an expert witness, usually they would take your word for it. And that's the whole point of having an expert in the courtroom. Someone who knows the field well can explain it to the jury and help them understand how the test works and why it's a viable test for developing evidence in the case. So it's an important function of the expert witness.